Guys, this is 101 Class Survival Guide, and this time I'm back with the next segment for nucleic acid sequencing, which is the next generation sequencing, also known as the high throughput sequencing. The contents for this video include the details about what is NGS and how it is very different from the other methods that we explained in the previous video, such as the Sanger's method, types of NGS methods, and the ones I'll be explaining include 454 pyro sequencing, Illumina Selecta sequencing, and PCR and its type. In this video, I'll be covering up to the 454 pyro sequencing method. So what is NGS or next generation sequencing, and why is it called high throughput sequencing? It is called so because it refers to a versatile technology of DNA sequencing that permits high throughput and highly adaptable whole genome scale assays at reasonable cost and high accuracy. The major similarity between NGS and the Sanger method is that both of these techniques are used for the DNA fragment sequencing. However, but in the NGS, it is massively parallel. Massively parallel means that this allows millions of fragments to be sequenced in a single run versus Sanger sequencing, which only produces one forward and reverse read at a time. There are a lot of NGS techniques, however, some of them include MPSS or massively parallel sequencing, which is a high throughput approach to DNA sequencing using the concept of massively parallel processing. MPSS is a procedure that is used to identify and quantify messenger RNA transcripts, resulting in data similar to serial analysis of gene expression. Polanyi sequencing is an inexpensive but highly accurate multiplex sequencing technique that can be used to read millions of immobilized DNA sequences in parallel. 454 pyro sequencing will be discussed next. Illumina Selecta will be discussed in the next video. However, it is a sequencing technique used to determine the series of base pairs in DNA, also known as DNA sequencing using the Illumina dye. Solid or sequencing by oligonucleotide ligation and detection is an NGS technology that generates hundreds of millions to billions of small sequence reads one at a time. DNA nanoball, also known as RCA or rolling circle amplification, is a high throughput sequencing technology that is used to determine the entire genomic sequence of an organism. The method uses rolling circle replication to amplify small fragments of genomic DNA into DNA nanoballs. The single molecule fluorescence sequencing is carried out on a glass flow cell with 25 channels for the same or different samples. The sample can be run with entire one or two flow cells at a time. The last one, the single molecule real-time sequencing, is a parallelized single molecule DNA sequencing method. The single molecule real-time sequencing utilizes a zero mode wavelength or ZMW. A single DNA polymerase is affixed at the bottom of a zero mode wavelength, which is a single molecule of a DNA as a template. This is used for the amplification. So these are some of the NGS techniques, and the one I'll be discussing in this video is the 454 pyro sequencing method. Okay, let's get into our first NGS technique, which is the 454 pyro sequencing. The basic working principle of the 454 pyro sequencing method is that it utilizes an emulsion-based sample preparation (EM-PCR) in which water oil droplets are used, in which the beads are present on which the massively parallel sequencing occurs. The working method is very simple, although it may seem complex, but it is very simple. Emulsion PCR uses small fragments of DNA, which are either restricted by chemical or sonication or any other method. They are then attached to small beads with the help of flanking regions A and B. I'll be discussing those in the next diagram. Compartmentalized into oil-water emulsified droplets. The small beads are then compartmentalized into the oil-water emulsions, in which they perform the massively parallel sequencing. Each droplet then acts as a single reactor. Like inside a single droplet, oil-water emulsified droplet, a single reaction occurs. An amplification process produces thousands of identical DNA fragments per beat through the PCR. The beads are freed from the droplets and introduced into a picotiter plate (PTB plate) with approximate capacity of read 1 into 10 raised to power 6 reads, each of about 400 base pairs in length simultaneously. Smaller beads containing enzymes for pyrosequencing are also introduced into each cell on the plate. Let's discuss this with a diagram. First of all, we require the sample input, which may be the genomic DNA, or BAC, which is the bacterial artificial chromosome, amplicon is our PCR product, and cDNA, which is the complementary DNA that is generated. Generation of small DNA fragments via nebulization, or we can use sonication. Ligation of A-B adapters flanking single-stranded DNA fragments. The single-stranded DNA fragments are each ligated to A-B adapters, which help them to bind to the beads. Emulsification of beads and fragments into oil-water microreactors. Clonal amplification of the fragments bound to the beads occurs in the microreactors. Now, this figure is really important and should be understood that after the clonal amplification has occurred, the emulsion is broken by the addition of isopropanol. The now double-stranded DNA is denatured, and the resulting beads carrying single-stranded DNA clones are deposited into the wells of fiber optic slide. Smaller beads linked to the enzyme ATP sulfurylase and luciferase are deposited into each well. As we can see here, that the oil-water emulsified droplet consists of the bead inside which the parallel sequencing has occurred. After we add the isopropanol, the droplet is dispersed and the beads are loaded into a PTP or picotiter plate. The PTP is placed inside a genome sequencer that detects the signals generated by each nucleotide addition. And the signals are in the form of light. And when each nucleotide is added, the results are noted and saved. Pyrophosphate generates signal upon addition of complementary nucleotide and is dark otherwise. When the signal is bright, it means that a complementary DNTP has been attached, and when it is dark, it means that no complementary DNTP has been attached. So this is how the 454 pyrosequencing method works. Thank you for watching. This is it for this lecture. And if you have any queries or future requests, please comment below.